We're going to be selectively having cells that are selectively permeable, bringing some things in and some things not coming across. They're excluded. They are said to be non-permeable. So if you have your generic cell, whether it's a red blood cell or any other cell in the body, if you have high potassium inside, potassium must be permeable. Most of the time, since the ATPase has been helping to get potassium high inside, potassium tends to leak out to where it's in lower concentration. And sodium will be high outside, and it tends to leak in where sodium is in lower concentration. Right now, we're going to stay talking about sodium and potassium. But very shortly, when we get to the nerves, things are going to change. But if you understand sodium and potassium permeability, then all you have to do is then find out where this next ion is in greatest concentration, and you can then figure out what's going to happen to that ion. So permeable, it can go across the membrane. Doesn't mean it has to go across a channel. It could be transported by an active transport in or out. It could be by a protein that is facilitating the diffusion in or out. So the plasma membrane being selectively permeable means not everything is going to go across the plasma membrane. So if it needs assistance, it is called facilitated or assisted diffusion or assisted transport. If it's happening all by itself, no protein involved, there's a channel there, but the protein isn't actually helping, it's just providing a hole for things to go through by the concentration gradient or electrical gradient, then it's unassisted. Active and passive transport can be assisted or unassisted. Well, actually active is requiring assistance because you're moving against a concentration gradient. But passive could be unassisted or it could be assisted, because you will have certain membrane proteins that will facilitate the diffusion of certain particles. Even though you are assisting something to go across the membrane, if that protein is not using energy to do that transport, it is a passive process. It's going to be using probably a gradient that is set up by active transport. So a lot of times they will call it secondary active transport. But if somebody's going again. So if we have a transport protein here, and it's using sodium's gradient, so it's binding sodium out here and then transporting something else across, using the concentration gradient of sodium to bring X in. This is down a concentration gradient, and it's bringing X in with it. No ATP consumed here. Yeah. No ATP consumed. So this is a passive process, but that concentration gradient being maintained by this ATPase over here that is consuming ATP and producing ADP. So this is happening secondary to what's going on over there. This is maintaining the concentration gradient for sodium. There's another transporter over here that binds Y, because there's a lot of Y's out here. So it's using the fact that there's a high concentration of Y out here to then bring Y into the cell. There is no ATPase that is maintaining Y's concentration inside at a lower concentration than outside. This is the variable. It either is high or it's going to be low. And usually it comes in here and it's consumed. And that's what's keeping that concentration low. This is facilitated transport, not referred to as secondary active transport, because there's no ATP involved in maintaining this concentration gradient. The gradient is, if you take this away, there's no gradient for it to come in. If Y is high out here and is transported in and consumed, that keeps the concentration gradient available, but it has to be high out here. So real life, you have glucose. When is glucose high?
After you eat.
After you eat. So shorthand for after, P with a line over it. After you eat, then glucose concentration goes up. Glucose goes into a cell. It's consumed by a lot of cells because they need it for energy. If it's not consumed, but it's linked together to form glycogen, then glucose disappears because now it's another molecule. So it's not necessarily being consumed, but it's being put into the glycogen molecule. So free glucose is kept low. So that's facilitated diffusion. Glucose can also come with sodium, and then it can be transported by secondary active transport. So lipid solubility, you don't go by size anymore. If it's water soluble, you're limited by bringing it across by a transporter, or if it goes through, it has some lipid solubility. The bigger a hydrophilic molecule is, the slower it comes in. In lipid solubility, the longer the carbon chain, the bigger the molecule, the more it's like that fatty acid tail on the phospholipid, so it goes through faster. So a little bit counterintuitive. Look back at your fixed diffusion slide. That is for water-soluble molecules, and so diffusion rate is inversely proportional to molecular weight. But remember, I told you for lipid solubility, the bigger the molecule, the higher the diffusion rate. So molecular weight is really in the numerator there for lipid solubility. This is the review. Nonpolar means there's very little charge on the molecule. It's not going to associate with water, which is a polar molecule. Since we are about 95% water, then we have a lot of water in our body that molecules may or may not associate with. If it doesn't associate with water, it's non-polar. Polar molecules will associate with the hydrogen and the oxygen in the water and then go into solution or suspension in the water. So an example of a nonpolar molecule is lipids, like the lipids in your bilayer. Why are they in the center? Because they don't like the water that's inside your cell and they don't like the water that's outside your cell. So the phospholipid that is amphipathic or amphiphilic or amphiphobic, once the phosphate head associates with water and the tails associate with each other in the center. Steroid molecules, they're made from cholesterol, which is a fat, so they're going to be lipid soluble. Something that is a protein that has a lot of amino acids and side chains, has a lot of charges on it. And something like glucose has charges on it, so it can associate with the water and go into solution. So if it's non-polar, goes right through the plasma membrane, doesn't really need a transporter. If it's hydrophilic, it's either going to be small enough to go through a channel or have enough of, or a limited number of charges that it can go with water through the lipid membrane, or it's got to be transported across. Unassisted have to go down a concentration gradient. So that could be through a channel, it could be through the plasma membrane. You've got lipid-soluble substances going across the plasma membrane, unassisted, just right through. And then you have some water-soluble that are also going down concentration gradients through a channel, like sodium going into the cell. For sodium and potassium, you've got this ATPase that's correcting for their diffusion that's happening down the concentration gradient. You also got an electrical gradient on proteins that are inside the cell that have net negative charges on them. So more proteins inside the cell than outside. They have more negative charges than positive charges, even after you cancel them out with the ions that are free-floating in there. Inside of the cell tends to be negative. So sodium has a concentration gradient and an electrical attraction to those negative charges. The sodium has two gradients driving it in. So driving force driving sodium, you've got a chemical plus an electrical gradient. For potassium, potassium's attracted to the negative charge, and it's got a concentration gradient driving it out. So for potassium, you've got chemical minus electrical. The electrical gradient wants to keep potassium in. The chemical gradient wants to allow potassium to go out. The sodium is going to enter more efficiently than potassium is going to exit. You come back up here to the ATPase that is correcting for this diffusion of potassium out and sodium in. 3 sodiums go out for every 2 potassiums that come in. Confirms that potassium is less likely to go out because its electrical gradient keeps it in than sodium is to come in because it's got chemical and electrical gradient pushing it in, driving it into the cell. So other ions come into play. You've got chloride. Chloride has a chemical gradient to go in, but it's running into all those negative charges inside, so it's going to be wanting to go out as well. It's going to be pushed out by its electrical gradient. So simple diffusion is unassisted. Simple means nothing else is involved but a concentration gradient. The Brownian motion, this comes from, I think, physics and chemistry use that term. This is a random movement of particles that aren't attached to other things. They will move based on their heat, their concentration, the volume that they have to move in. They will hit when they're moving quickly because they're in high concentration, bouncing off each other, or you have a limited space for them to move in, so they're bouncing off the the whatever container they're in and each other. If you place a semi-permeable membrane into these moving particles, when they hit the membrane, they have a chance to diffuse across. Maybe they bounce off, but more than likely, if they're soluble in the membrane, they're going to go across. If they're small enough, they're going to get trapped in the membrane and work their way across. Heat up the particles, they move faster, more likely to hit that semi-permeable membrane and go across. So the higher the concentration, the more likely they're going to go across. Lower concentration, less contact with the membrane, so less likely to go across. Even though you're going down a concentration gradient, there is going to be diffusion out of across in the opposite direction. So sodium is more likely to go in, but there are going to be some sodiums that are going to go out. Potassium tends to go out, but there are going to be some potassiums that will go back in. Just when they hit the membrane and go across is going to be determined by their concentration on either side of that membrane. The higher the concentration, more likely they're going to hit the membrane and go across. If you didn't have ATPases correcting for this diffusion, sodium would reach its equilibrium and potassium would reach its equilibrium. And then net diffusion would cease. So still diffusion happening, but every time one goes across in one direction, another one will go across in the opposite direction. Our body, every cell is relying on concentration gradients to survive. It's not going to survive if the concentration gradient goes to equilibrium. No cell in your body is going to survive that. So one way that you cause cells to go to equilibrium is poison the ATPase. So some pesticides and some lethal gases and things, they'll poison the ATPase so cells run to equilibrium and the animal or the person dies. Here we've got no permeable membrane, just high concentration to the left, low concentration to the right. Same particles, but lower in B, higher in A. There is going to be diffusion to the left from lower to higher, but net diffusion is going from left to right. You let those molecules move randomly. More molecules are going from A to B than from B to A. When you get to the equal dispersion of all the molecules, you have reached equilibrium. So now there's still diffusion happening, but when one molecule goes to B, another one goes back to A. Reading the chart, the bigger the arrow, the greater the gradient for movement. Smaller arrow means less movement in the opposite direction. Equal size arrow is telling you you've reached equilibrium. Oh, we didn't get to this. No wonder people kind of look at me like, go back to what slide? Okay, so here on this, this is Fick's law of diffusion. It's telling you what is important for simple diffusion. Most important is going to be your concentration gradient. For biological situations, you're not usually, in normal situations, you're not changing surface area. You're not changing membrane thickness from time 1 to time 2. But what you are changing is concentration of a solute. So after a meal, you have a lot of glucose. suddenly glucose is available for diffusion into cells. The surface area of the membrane, if you want a lot of diffusion, you have to fold the apical plasma membrane, make microvilli there so that there's more available surface area for diffusion to happen. And even on the lateral membranes and the basal membrane, if you want a lot of movement across those plasma membranes, you have to fold them as well. So usually microvilli are for apical plasma membranes, but you can have infoldings and a lot of foldings laterally and basally as well. Higher the lipid solubility, faster diffusion is going to happen. Why? Because 95% of your plasma membrane is lipid. So you've got 95% of the surface area that's available for lipid to go across. 5% are going to be other proteins. Some of them may be channels. Some of them may be other proteins that are not allowing for hydrophilic things to go across. So that limits hydrophilic diffusion. If you increase the distance that the molecule has to go across, it's going to take it longer to get across. It's very intuitive. You make something thicker, it takes longer for something to go across that thick membrane than a thin membrane. Why do people have problems with blue lips and fatigue, not able to exercise when they have pneumonia? Because fluid collects in the lungs, and now your gases have to go across the fluid plus the diffusion barrier. You make the diffusion barrier thicker, then oxygen doesn't go across. If oxygen doesn't go across, energy production goes down in your mitochondria, and so you're more easily fatigued. Smoke a lot, your basal lamina thickens because of all the pollutants and the irritants that you're sucking in, so it's going to create a long-term change. You can cure the pneumonia. You can't really cure the basal lamina thickness from smoking. Other things tend to remodel, but it's highly unlikely that that basal lamina is going to be remodeled. So you have an equation here. Net diffusion is related to the change. Delta means change. Big C's concentration gradient. A is your surface area, and B is the lipid solubility over the square root of the molecular weight of the substance times the distance that the molecule has to diffuse across. So this is an equation for the diffusion of a lipid-soluble substance. Molecular weight, so what this is saying is diffusion is inversely related to molecular weight. That is not true in a lipid-soluble substance. So nonpolar, the molecular weight has to be up top with lipid solubility. Just understand the relationship there. The diffusion constant, we'll talk more about that later. Permeability is related to the diffusion constant over the membrane thickness, so it would be more important next semester. So again, nonpolar, you have 95% of that plasma membrane for nonpolar substances to go through. So it's easy for those to just dissolve in the lipid and diffuse either in or out of the cell. Small ions can follow water through the plasma membrane, but if they're important, like sodium and potassium, they'll have their own channels that they can move in as well. When the channel is always open, it's referred to as a leak channel. So these two channels are leaking sodium and potassium constantly in and out. There are other channels that are voltage gated. That means you have to have some increase in charge, positive charge inside the membrane, and the decrease in positive charge outside the membrane to reverse what you started with. So right now the cell is negative inside with respect to outside, so outside is positive. If you reverse the charge just right at the plasma membrane, you have positive charges accumulating here, leaving negative charges outside. Then you can open channels that will allow a lot of sodium to go into the cell. Or you can open channels that will allow a lot of potassium to leave the cell. You could also do something to other channels, like calcium channels and chloride channels, that we'll talk about when we get into the nervous system. So you have leak channels, and then you have voltage gated channels. You might also have a ligand gated channel that is closed until a neurotransmitter or a hormone binds, and then your ion can move. So semi-permeable membrane, that's what the dashed line means. Some things go across, some things do not. What is the universal solvent for the human body? Water. So think always these solutes are in water. If solute can move, water can move. Even when solute cannot move, if it's a semi-permeable membrane, it's going to be permeable to water. Water always moves first. So here it's a little bit incomplete. We've got solute here. What is it in? Water. We've got solute here. What is it in? Water. What's the first thing that's going to move? Water. So the arrows saying that solute is moving. That's the second thing that happens. The first thing is water moves. Where is water going to move? From the right to the left, or the left to the right? From the right to the left. And after it reaches its equilibrium, then as it's approaching its equilibrium, then these heavy solutes start to go in down their concentration gradient. So water moves first, and then solute moves. Solute doesn't care about water, it only cares about its concentration. And water doesn't care about solute, it only cares about its concentration. So more water here, it moves to the left. More solute here, it moves to the right. Okay, this says no diffusion occurs. Right or wrong? Wrong. You're supposed to say, wrong! What moves? Where does it move? Towards, don't mess your minds. Look, where is water in the highest concentration? On the right. Why? Because there's less solute. Separate those out. Don't say water's moving to where solute's in higher concentration. Say water's moving to where water's in lower concentration. Why is it in lower concentration? Because there's more solute. So separate those out. I guarantee you, if you don't, during an exam, you're going to mess up. So draw the picture too. If I say you have a semi-permeable membrane, chambers A and B, between A and B, then draw a little beaker for yourself and put a dashed line to get your semi-permeable membrane. And then I tell you, okay, in so draw your beaker, put your semi-permeable membrane, put A, put B. Draw that first and then read the rest of the question. So I say in chamber A, you have a 2 molar solution of sodium chloride. And in B, you have a 2 molar solution of glucose. Where did I determine this, the 2 molar? When I added it to the water or before I added it to the water? Before. What does 2 moles mean? What is a mole? It's a measurement. Which of these molecules weighs more? Glucose. Glucose weighs 180 and sodium 58.4 grams per mole. Do I have different numbers of particles? No, I do not. I have 1 mole of each of these types of particles. 1 mole is equal to 6.023 times 10 to the 23rd particles. What is that number? Avogadro's number. So we have the same number of particles per mole. One weighs more. It's a bigger, has more carbons in it, for one thing, than sodium chloride, which has no carbons. So this weighs 58.4 grams per mole. Glucose, 180 grams per mole. Both moles contain 6.023 times 10 to the 23rd particles. That's dry. So I have my little scale and I put my particles on there and I weigh out a mole. And the dial goes up and goes 180. So I dump that into my beaker, my flask, and then I fill it up to the liter line with H2O. So if this is glucose, and this is sodium chloride, oops, I have a mole of glucose dry, and then I fill it up to the liter line, a mole of sodium chloride dry, and then I fill it up with water. I had 2 moles of glucose. I had 2 moles of sodium chloride. Are they going to be in equal particle concentrations when they hit water?
Concentration.
Which of these particles, what kind of bond is it here? Ionic. What kind of bond is it here? Covalent. Which of these comes apart when you put it into water? Ionic. Oh boy, pressing buttons again. Okay, so ionic bonds will disassociate. You'll get sodium and chloride. You're going to end up with 2, if you get complete disassociation, you get 2 moles of sodium and 2 moles of chloride from this dry crystal of NaCl. Oops. I went backward instead of forward. Okay. Here, covalent bonds can't come apart, right? So you have 2 moles dry, you have 2 moles wet. Here you've got around 4 moles wet. So, where is water in higher concentration? It's high here because we still only have 2 moles. Now this has turned into 4 moles. So we have a low water concentration here, high water concentration on side B. What's going to happen first?
Water's going to move.
Water moves in which direction? It's going towards A. So on the side here, say water, number 1, water moves from B to A. If I told you that the sodium, so we got black now. Okay. Sodium is permeable, but chloride is not. So sodium can cross, but chloride, draw a little arrow like that, like they had on the previous slide. It's got a curved arrow, meaning it can't cross. If I told you sodium can cross, chloride can't, what do you think about glucose? Haven't told you about glucose, but what do you infer from the fact that chloride cannot go across? Probably glucose is not going across either. If I told you the membrane was charged negatively, then it could be that chloride is not going across because it's being repelled by the negative charge on the membrane. But I didn't say that. Most of the time, if the small particle goes, this huge particle is not going. So I might tell you about glucose, I might not tell you about glucose. But you have to read your choices and then see what comes out to be the best answer. The sodium is permeable, chloride is not. Is sodium going to move before glucose? If glucose could move, which is going to move first? The sodium, because it's smaller. So the smaller the molecule and also the higher concentration, we've got 2 moles of sodium that are now bombarding this semi-permeable membrane. You got 2 moles of glucose, but moving slower. So who's going to bombard the membrane more? It's going to be sodium. So water moves first from B to A. Second thing that's going to happen is sodium. Sodium's going to move from A to B, down its concentration gradient. There's 0 sodium on this side, so I've got a huge concentration gradient to go down. Okay, so those things you know for sure. You don't know if glucose can go, but if water leaves and sodium goes in, what's happening to glucose concentrations? So number 3, you could just look at glucose and its concentration. What's happening to the concentration of glucose? If water leaves and sodium comes in, is it concentration going up or down? Okay. Okay, it's going up because water leaves, and it's going down because sodium enters, right? Because sodium is getting in between the glucose molecules. So you don't know. 1 mole of, you got water moving, 1 mole of sodium going over, but you don't know exactly what's happening to the glucose concentrations. So what about chloride? What's happening to the concentration of chloride? Okay, what happens initially when water comes over? It goes down. What happens when sodium leaves?
It goes down.
Increases.
It increases. So you don't really know about chloride either. It's not really clear. And it has to be, I can't have you spending all this time thinking, well, how much water came over and then how much sodium left and all this. It has to be really clear or I'm not going to ask it. Okay. 5. What happens to, if I said that, like towards the end, water moves from A to B. So you said this is true, this is true, these you don't really know for sure. If I had a statement that water moves from A to B towards the end of diffusion, is that a true statement?
No, it moves first. Water moves first.
Oh, but you're not thinking far enough. Okay, water moves from B to A first. Water always moves first. But sodium went from A to B. So what is that doing to water concentration here now? It's decreasing water concentration here. So some water is going to follow that sodium back into chamber B. So this would be a true statement that towards the end, water would move those permeable sodium, follow those permeable sodium ions because they're changing the water concentration in B. So you look for the true statements. If there's 3 true, there's probably a combination in choice D and E, whether A and B are true or A, B, and C are true, something like that. Okay, time to take a break.